Hello guys, good morning. I welcome you all on Unacademy, India's largest learning platform. Myself, Dr. Nidhi, I'm your physiology educator at Unacademy. This is our Telegram page. You can join us out there. This is my page on Unacademy, that is unacademy.com/@KGMC. Follow me out there so that you'll get notification of any upcoming classes of mine on Unacademy app. This is the code Dr. Nidhi. If anybody wants to join any paid course, you can use this to get additional 10% off. I've taken many plus classes, many special classes, so anytime you can join and revise your topic. Now coming to the subscription, we have this plus subscription. What all are the benefit? You will be able to access both the live and recorded session. By chance you miss a session, you can see the recorded version. You have an opportunity to learn from India's top educator for the medical examination. We always conduct this live test and live quizzes. From that you can analyze your preparation at any point of time. You have a flexibility, you can study on the device of your choice. You always carry your phone with yourself, so you can access your class when you are anywhere. We have a question bank which consists of 25,000 plus questions. These questions are high yield, clinical based and with explanation. So this is going to be very beneficial for all of you. Next is the iconic subscription, where you get access of India's two best platform, that is our Unacademy and the Prep Ladder. So you are going to get all the plus benefit. Like I said, live classes, recorded classes, question bank, live test, live quizzes and the PDF. Along with that, you get access for this Prep Ladder. In this, you can access the clinical and integrated essential of Prep Ladder, video lecture from the Dream Team, Question Bank 3.0, rapid revision and snapshot, and you will also be provided with the treasure and dream notes. So if you are looking for the subscription that include both, go for the iconic subscription. Special class feature, our special class is also free of cost, but they are way more special because they are always live interactive sessions are there. Polls feature are there by which you can better understand your topic. Raise a hand features there. Now you can talk to educator in the live session. If you follow any educator, you will get notification of their upcoming classes. After the lecture, you can download the PDF and revise it anytime, anywhere. Important announcement, we are started with this unlock 20, जहाँ पे आपको subscription में 20% off मिलेगा by using the code that is Dr. So do enroll yourself right now to avail this offer, and this offer is till 30th June. And we have started this batch, that is target NEET PG 23 batch, that consists of a 2 module. Module 1 में subject wise batch होगा, and module 2 में we are going to revise all those. So next batch we started out for the repeaters, that is NEET PG 23 test and discussion batch for the repeaters. So as per your requirement, you can take your subscription and start your preparation right now. So important announcement, iconic price subscription का भी price hike होने वाला है. So if you are looking for the iconic subscription, do enroll yourself right now so that you can save your extra penny. And this is the code Dr. Nidhi, by which you will get additional 10% off. Okay, then coming to our topic, today we are going to study about the nerve action potential or the nerve physiology. So first, coming to this action potential, first of all, understand all the cells in our central nervous system. So, two types of cell basically present in our nervous system, that is the neuronal cell and the glial cells are there. First, coming to the neuronal cell. Neuronal cell can be either unipolar, bipolar, multipolar or pseudo-unipolar. And the glial cell can be either macroglial, which are larger in size, and the one which is smaller in size is known as microglial cells. In a macroglial cell, we have this astrocyte, oligodendrocytes and the Schwann cell, and in microglial cell, we have this phagocytic cell. Now coming to the detail of each and every glial cell first of all. So first coming to this astrocyte. This astro stand for astro. This stand for the star shape, because the shape of this cell is star-like. So it is known as astrocyte. Basically two type of astrocyte is there. Fibrous astrocyte as well as protoplasmic astrocyte. This is the fibrous astrocyte. Okay, and this one is the protoplasmic astrocyte. So the first coming to the fibrous astrocyte is present in our white matter, while this protoplasmic astrocyte is present in the gray matter. What is the function of this astrocyte? It forms this blood-brain barrier, And what does it do? It has nutritive role to the neurons. It uptake the nutrition and it sends to the neuronal cell, and it uptake the potassium ion and neurotransmitter, and that is also sent to the our neuronal cell. So basically what happened, it is playing this nutritive function. Okay, is this clear with everybody? The astrocyte. Now coming to the oligodendrocyte and Schwann cell. These two cells both are responsible for myelination. The oligodendrocyte is responsible for central nervous system myelination, while the Schwann cell is responsible for peripheral nervous system myelination. Okay, so this oligodendrocyte is responsible for multiple myelination. Means one oligodendrocyte is going to form myelin sheath around multiple neuron. And basically, अगर ratio की बात करें तो its ratio is approximately 1:30. Means one oligodendrocyte myelinate approximately 30 neurons. So this is the characteristic features shown by our oligodendrocyte, that is multiple myelination. Now coming to the Schwann cell. Schwann cell are responsible for peripheral nervous system myelination, but they myelinate only around a single nerve fiber. So this point is important. Do remember, oligodendrocyte is responsible for central nervous system myelination. Schwann cell is responsible for peripheral nervous system myelination. See, this is our oligodendrocyte. This oligodendrocyte is responsible for myelination of this neuron, and this is responsible for central nervous system myelination. Okay, now is the Schwann cell. Schwann cell is responsible for peripheral nervous system myelination, and it myelinate around single neuron. So this is the basic difference between these two. Now coming to the neuronal cell. Neuronal cell can be either unipolar, pseudo-unipolar, bipolar, or multipolar. So now coming to the point, what do you mean by this unipolar, pseudo-unipolar, or the bipolar? Let me show you the diagram. When a single structure arises from cell body, it is known as unipolar. When two structure arises from cell body, it is known as a bipolar. Next is the pseudo-unipolar. It looks like a unipolar, but it is not actually unipolar. Initially, only one structure arises from cell body. Later on, it divides. This type of neuron is known as pseudo-unipolar. Next is the multipolar. When multiple structures are arising from our cell body, it is known as multipolar. Is this clear with everybody? Now the examples are important. The unipolar neuron are present in the invertebrates. Pseudo-unipolar neuron is present in our dorsal root ganglion. Bipolar neuron is present in our retina, olfactory epithelium, sensory glial of cochlear and the vestibular nerve. And the multipolar neuron is present in our spinal motor neurons. Okay, so the examples are very, very important. Do remember? So now moving further to the parts of neuron. To understand this part, let me draw a basic diagram of our neuron. This is basically the cell body. Okay, these are the dendrites. The cell body consists of nucleus, which consists of nucleolus. Okay, it consists of a Golgi bodies. It consists of this Nissus granule, which is nothing. It is our rough endoplasmic reticulum of our neuron. Okay, and in myelinated neuron, we have this myelin sheath. Okay, so first is the perikaryon, soma, or cell body. This is perikaryon, soma, or cell body. This consists of nucleus, this consists of Golgi apparatus, this consists of a Nissus body. Do remember the Nissus body are the rough endoplasmic reticulum of our neuron. Do remember, this is important from the exam point of view. Next is the dendrites. Dendrites, these are the dendrites. They are known as the receiving end because it receives the signals, and it is also known as the receptor zone. Next is the axon hillock. As the name suggests, it is very confusing from the name that it is a part of axon. No, it is not a part of axon. It is a part of cell body jahan se hamara axon arise kar raha hai. So this is our axon hillock. So axon hillock is not a part of axon, it is a part of cyton from where the axon arises. Is this clear? Next is our initial segment. This is the first unmyelinated portion of axon. So see, this is the diagram. This is the first let me change the color for all of you. This is the first unmyelinated part of axon. It is known as initial segment. This axon hillock and the initial segment is collectively known as a trigger zone. Why it is known as trigger zone? Because out here the concentration of voltage gated sodium channel is more. So the threshold of these 2 points are very low. So they are the site of generation of action potential. Okay, is this clear? So that's why they are collectively known as a trigger zone. So first coming to this initial segment, what do you mean by this initial segment? It is the first unmyelinated portion of axon and it has lowest threshold. And the last is the axon telodendria, which is also known as synaptic knobs or the terminal buttons. Okay, so these are the axon telodendrias. Is this clear? So now moving to the important point, the conduction speed depends upon the myelin sheath presence. More the myelin sheath, more will be the conduction. So myelinated neurons have faster conduction as compared to unmyelinated neurons. Okay, this point is also important. Myelination is dependent upon, conduction speed is dependent upon the presence of a myelin sheath. So this is the diagram, same diagram which we drawed. So first is the cell body or the soma or the perikaryon. It consists of this nucleus. This nucleus consists of this nucleolus. Next is this dendrite, which is also known as the receiving end because it receives the signal. Okay. Next is the axon hillock. It is the part of axon, part of cyton जहां से हमारा axon arise करता है. Next is the initial segment. Initial segment is the part of axon. First unmyelinated part of axon is the initial segment and they collectively known as a trigger zone. Why they are known as trigger zone? Because the concentration of voltage gated sodium channel out here is high. That's why threshold is lower. So this is the site for generation of action potential. So it is known as trigger zone. Next, the axon is there. Okay, these are the myelin sheath. Okay, these are the myelin sheath which is formed by the Schwann cell, and these are the internodal region. The portion of axon which is not having this myelin sheath is known as node of Ranvier, and the two node of Ranvier, the distance between two node of Ranvier is known as internode. Okay, is this clear? So this is the synaptic knobs you can see in this diagram. These are all the synaptic knobs. So now coming to the node of Ranvier. We already discussed in the diagram, the area of axon which is devoiding of this myelin sheath is known as node of Ranvier. And this node of Ranvier is also having high concentration of voltage gated sodium channel. So they are also the site of action potential for the sensory neuron. Okay, so do remember this one. This is very, very important. For the sensory neuron, the site of action potential generation is first the node of Ranvier, while for the motor neuron, it is the initial segment. Is this clear with everybody? Do reply fast. Is this clear? If anybody has any doubt till now, you can ask. Is this clear? This point is very, very important. Good morning, Dr. Hirsch. Is this clear with everybody? Shall we move further? Okay then, moving further. Now coming to the exoplasmic transport. This exoplasmic transport is in two directions. First is known as anterograde transport. Another one is known as retrograde transport. Now coming to the point, what do you mean by this anterograde transport or the retrograde transport? The transport that is occurring from cell body to the axon is known as this anterograde transport, and the transport that is occurring from our axon to cyton is known as a retrograde transport. This anterograde transport is done by the dynein molecule. Do you remember this point is important? And this retrograde transport is done by this kinesin molecule. Okay, so these all are the microtubule. If you studied about the cytoskeletal filament of our cell, the microtubules are there, like dynein, microtubulin, and the kinesin. The anterograde transport is done by this dynein, and the retrograde transport is done by the kinesin. Okay, is this clear? Now coming to the action potential of our neuron, or you can say electrical event in our neuron. This electrical event in our neuron is recorded by cathode ray oscilloscope. To understand this, first understand some important terminology. First is the latent period. What do you mean by this latent period? The time gap between the stimulus and the response is known as the latent period. And to get the response, how should be the stimulus? Intensity of a stimulus should be rapidly rising. This point is important because slowly rising stimulus has higher chance of accommodation, and duration of stimulus should be optimal enough to produce the response. Okay. Next is the threshold stimulus. What do you mean by this threshold stimulus? Neuron only and only respond to this stimulus. Any supra-threshold stimulus or the sub-threshold stimulus is not going to produce any response. Our neuron only respond to this stimulus. Is this clear? Everybody, is this clear? Okay then. Now coming to this strength duration curve. On y-axis, we take the stimulus duration. On y-axis, we take this stimulus strength. In this, we have two terminology. What first is the Rio base, second one is chronaxy. What do you mean by this Rio base? What do you mean by this chronaxy? Rio base is the minimum strength of stimulus that is required to produce the response. Okay, what do you mean by this Rio base? Rio base is the minimum strength of stimulus that is required to produce this response. Now, what do you mean by this chronaxy? Chronaxy is the time taken to get the response when the strength of stimulus is twice of the Rio base. Is this clear? What do you mean by Rio base? What do you mean by chronaxy? Everybody? Rio base is the minimum strength of a stimulus that is required to produce a response, and chronaxy is the time taken to produce a response when the strength of a stimulus is twice of a Rio base. Okay, so same thing is written out here. The Rio base is the minimum strength of stimulus that is required to produce a response. What do you mean by this chronaxy? Chronaxy is the time taken by tissue to produce a response when stimulus strength is double the Rio base. Okay, Rio base normal होता है 1 milliampere and chronaxy is normally 1 millisecond. So we can say the more the time taken by the tissue to produce response means tissue is less excitable. So we can say chronaxy is inversely proportional to the excitability. जितना ज़्यादा time लगेगा उतना less excitable है हमारा tissue. So this is the increasing order of chronaxy, or you can say decreasing order of excitability. Okay. So सबसे ज़्यादा excitable क्या होता है हमारा myelinated nerve, then comes unmyelinated nerve, then come our skeletal muscle, then comes our cardiac muscle, then comes our smooth muscle. Now there was another two terminology, that is our accommodation as well as adaptation. What do you mean by these two? We studied that neuron only respond to the threshold level. The sub-threshold level stimulus is also not going to produce response. Supra-threshold stimulus is also not going to produce any response, or no action potential will be generated in that situation. Why is it so? Sub-threshold stimulus is going to cause this accommodation, and that's why no action potential is generated. Supra-threshold stimulus is going to cause adaptation in our neuron. So that's why no action potential will be generated. Is this clear, everybody? What do you mean by this term accommodation and adaptation, everyone? Do reply fast. Is this clear? Okay, then moving further. Now coming to the different phases of action potential. So first phase is this one, which is known as local potential change. This is the first phase of action potential of our neuron. Second phase of action potential of our neuron is this repolarization phase. Third phase is our repolarization phase. And the fourth phase is our hyperpolarization phase. So first coming to the local potential change. We all know the resting membrane potential of our neuron is, RMP of neuron is minus t millivolt. Okay, and due to local potential change, why this local potential change is there? Because of slow influx of a sodium channel. Not sodium channel. So slow influx of the sodium is going to cause this local potential change. Since the positive ion is moving inside the cell, we all know membranal negativity is going to decrease, and from minus 70 it reaches to minus 55 millivolt. This minus 55 millivolt is also known as a firing level or the threshold potential. Okay, why it is known as threshold potential? Because when membranal potential reach to this minus 55 millivolt, the action potential will be generated, which is including our depolarization plus repolarization. ठीक है, जैसे ही हमारा मेम्ब्रेन पोटेंशियल माइनस 55 मिलीवोल्ट पे पहुंचेगा, उसके बाद ही एक्शन पोटेंशियल जनरेट होता है. दैट्स व्हाट इज नोन एज थ्रेशोल्ड पोटेंशियल ऑफ फायरिंग लेवल. Next phase is the depolarization. Depolarization is also due to sodium influx. And due to sodium influx, membranal negativity is going to decrease and membrane का potential overshoots करता है to plus 35 millivolt. Okay. So the membranal potential overshoots to plus 35 millivolt due to depolarization. Now the repolarization is due to potassium efflux. And since positive ion is moving in outside the cell, membranal positivity is going to decrease. So it decreases from plus 35 millivolt to minus 55 millivolt. Okay, and after that what happened, this potassium channel is closing very slowly, so there will be the continued potassium efflux, and due to continued potassium efflux, what happened, the membranal negativity is going to increase, and it increases beyond minus 70 millivolt, and this phase is known as hyperpolarization phase. Is this clear with everybody? So we have four phases: local potential change, depolarization, repolarization, and the last one is hyperpolarization. See, all these are written. In phase one, local potential change is there, and this is due to slow sodium influx. And due to that, the membranal potential from minus 70 reaches to minus 55 millivolt. And this is known as threshold potential or the firing level. Next is the phase 2, which is known as a depolarization phase. And it is also due to sodium influx. And due to that, potential overshoots to plus 35 millivolt. Next is the repolarization phase. This repolarization phase is due to potassium efflux, and that's why what happened, membrane potential is going to reach back to its resting potential, that is minus 70 millivolt. But when membranal potential reaches minus 70 millivolt, there is slow return of this potassium channel to the closed state, and there is continued potassium efflux for a certain period of time is there. And due to that क्या होगा, membranal negativity is going to increase, and it increases till minus the 90 millivolt. And this hyperpolarization phase is going to cause inhibition of neuronal function. This is the significance part. Is this clear with everybody? Do reply fast. Is this clear? If anybody has any doubt, you can ask. Now moving further, now coming to the clinical correlation part. Inhibitory neurotransmitter like our GABA, how they work? They either causes chloride influx or the potassium efflux. What happened? This is our cell. When chloride influx is there, membranal negativity is going to increase. When potassium efflux is there, membranal negativity is going to increase. And that was responsible for the neuronal inhibitions. That is going to cause this hyperpolarization phase. And the hyperpolarization phase is going to cause neuronal inhibition. And that's how our GABA works. Okay. Next, the GABA agonist. GABA agonists like barbiturate or benzodiazepine, they are also going to cause the hyperpolarization phase, and that's why they are going to cause the sedations. Is this clear? Now coming to the next concept, that is a refractory period. This is very important. Two type of refractory period is there. Absolute refractory period or the relative refractory period. What do you mean by this absolute refractory period or the relative refractory period? First, coming to this absolute refractory period. It is the period in action potential in which a cell is unable to fire second stimulus if any stimulus of any strength, any duration is given. ठीक है, कितने भी स्ट्रेंथ का स्टिमुलस, कितनी भी ड्यूरेशन का स्टिमुलस देंगे, इस पीरियड में हमारा सेकेंड एक्शन पोटेंशियल विल नॉट बी जनरेटेड. व्हाइल रिलेटिव रिफ्रैक्टिव पीरियड में क्या होता है, अगर रिलेटिवली मैक्सिमम स्ट्रेंथ का स्टिमुलस देंगे तो सेकेंड एक्शन पोटेंशियल विल बी जनरेटेड. ठीक है, अब बात करेंगे एब्सोल्यूट रिफ्रैक्टिव पीरियड स्टार्ट्स फ्रॉम एक्शन पोटेंशियल टिल 1/3 of a repolarization start of action potential to till 1/3 of repolarization is our absolute refractive period. In this period, any strength of a stimulus or any duration of a stimulus is not going to produce a second action potential. Next is the relative refractive period. It starts from 1/3 of repolarization till the end of action potential. In this, when we give the maximum strength stimulus, second action potential will be generated. So we can say this one, longer the refractive period, there will be lesser the frequency of stimulation will be there. Is this clear, everybody? Longer the refractive period, frequency of stimulation will be lower. So see, this is the diagram. Let me correct this one. This period. So this is the till 1/3 of repolarization. This is our absolute refractive period. Okay. Next is the relative refractive period. So this is our relative refractive period. Till the end, 1/3 of repolarization till the end of action potential is our relative refractive period. Is this clear with everybody? In relative refractory period, अगर maximum strength का stimulus देंगे, second action potential will be generated. Is this clear, everyone? Shall we move further? Okay, then moving further, now coming to the gate concept that is applied for our voltage gated sodium channel. According to the gate concept, this voltage gated sodium channel consists of a two gate. Suppose this is our voltage gated sodium channel. It consists of this two gate. Outer gate is known as M gate or the activation gate. Okay, the inner gate is also known as H gate or inactivation gate. Okay, how they work? On the basis of opening and closure of these two gates, there is three possible state of voltage gated sodium channel. They are, first is the resting state or the closed state. This is the diagram of a resting state. In resting state, what we all are seeing is M gate is closed. Means our activation gate is closed and our inactivation gate H gate is open. तो यहाँ पे सिर्फ activation gate is closed. Next is the activation state or the open state. In this, both gates are open, and due to that, what happens? Sodium influx occurs during the depolarization phase. And you can see this activation gate is seen during the depolarization phase of our neuron. Next is this inactive state. In inactive state, क्या होगा? Inactivation gate is closed. Okay, so this inactivation gate or H gate is closed while this M gate is open up. And this is seen during the repolarization phase. So these are the three possible state of voltage gated sodium channel on the basis of opening and closure of this M gate and H gate. Okay, so this is the gate concept for the voltage gated sodium channel. Okay, everybody? Is this clear? If anybody has any doubt till now, you can ask. Do reply fast, everyone. Is this clear with everybody? Shall we move further? Now we are going to see the application part of this voltage gated sodium channel. So what happened? Local anesthesia has lower affinity of sodium channel when it is in the resting state. It has higher affinity towards the inactivated state of voltage gated sodium channel. So this local anesthesia is going to bind with the sodium channel when it is in its inactivated state. So it is going to bind and prolong the inactivated state of voltage gated sodium channel. So that's how our LA work. Okay, is this clear with everybody? Now next we are going to see questions related to our today's topic. So coming to the first question of today's section. Non-myelinated axon differ from myelinated one in that they are lacking node of Ranvier, they are more excitable, are capable of regeneration, are not associated with the Schwann cell. So everybody answer this question fast. Non-myelinated axon differ from myelinated one in that they are more excitable, lack node of Ranvier, are capable of regeneration, are not associated with the Schwann cell. Everybody answer this question fast. This was the easy one, I guess. They lack node of Ranvier, and non-myelinated neuron are less excitable. Okay, they are not capable of regeneration because myelin sheath is absent. And do remember, this point is very important. Schwann cell is present in both myelinated as well as non-myelinated neuron. Okay, so answer is only and only B. Is this clear with everybody? Coming to the next one, synaptic potential can be recorded by patch clamp technique, voltage clamp technique, micro electrode, or the EEG. Synaptic potential can be recorded by. Everybody answer this question fast. The potential at the level of synapse can be recorded by. It can be recorded by using the microelectrode. One electrode is placed at the presynaptic membrane and another at the postsynaptic membrane. Okay, coming to the next one. Magnitude of action potential is determined by sodium, magnesium, potassium, or the calcium. The magnitude of action potential is determined by. Everybody answer this question fast. Magnitude of action potential is determined by sodium ion. More the influx of sodium ion, more will be the action potential generated. Coming to the next one. Action potential is initiated at axon, hillock, or the initial segment of neuron void. Option A, threshold for excitation is the lowest. Neurotransmitter is released at the site. It is an unmyelinated segment, has the lowest concentration of a voltage gated sodium channel. Action potential is initiated at axon, hillock, or initial segment of neuron. Why is it so? Everybody answer this question fast. Action potential is initiated at axon, hillock, and initial segment, which is known as trigger zone, because the threshold out here is the lowest. Why the threshold out here is lowest? Because out here the voltage gated sodium channel is more. Answer is A. No, they are the undermyelinated segments, but question is asking why the action potential is generated out here. Okay, this statement is also true. Undermyelinated segments they are, but threshold for excitation is the lowest. That's why it is the sites where action potential is initiated. Coming to the next one. Synaptic conduction is mostly orthodromic because dendrite cannot be depolarized. Once repolarized, an area cannot be depolarized. Strength of antidome impulse is less. Chemical mediator is localized only in the presynaptic terminals. Everybody answer this question. Synaptic conduction is mostly orthodromic. Why is it so? Everybody answer this question fast. Synaptic conduction कैसा होता है? Unidirectional होता है. Why is it so? Because the chemical mediator is localized in the presynaptic terminal and its receptor are only and only present at the postsynaptic membrane. ठीक है? That's why the synaptic conduction is only unidirectional. ठीक है? Is this clear with everybody? Do reply fast. Is this clear? Okay, then coming to the next question. True about the nerve impulses travel in one direction along axon. If current is increased, too slow nerve respond fast. Option C, travel in one direction at synapse, travel with the speed of electric current. True about the nerve impulse is, I guess this is the easy one. Travel in one direction along axon. If current is increased, too slow, nerve respond fast. Travel in one direction at synapse. Travel with the speed of electric current. Yeah, very true. At the level of synapse, travel in one direction. Coming to the next one. Unidirectional flow of nerve impulse is at synapse, axon, dendrite, all of them. We already done this one, so skipping this one. Coming to the next one. This is a nice question. True about the Nessels granule. Involve in RNA synthesis, present in axon, present in dendrite, present involved in protein synthesis. Structurally, they are endoplasmic reticulum. So this basically consists of a multiple answer. So see each and every option very correctly. True about a Nessels granule. Everybody answer this question fast. Nessels granular, rough endoplasmic reticulum of our neurons. So they are responsible for RNA synthesis, they are responsible for protein synthesis, and they are present in our dendrite region. So answer is A, C, D, and E. Is this clear with everybody? Coming to the next one, rapid axonal flow in the neuron is mediated by all except dynein, kinesin, neurofilament, or the microtubule. Rapid axonal flow in the neuron is mediated by. Answer this question fast, everybody. Yeah, the neurofilament is not required for our axonal transport. Do you remember अपने सेशन में भी बात की थी, dynein, kinesin, and microtubule are responsible for axonal transport. See, this is the diagram also. Anterograde transport required kinesin and retrograde transport required dynein. These are the microtubules. So neurofilament is not required for our axonal transport. Coming to the next one. Axonal transport is interrograde, retrograde, both or none. Axonal transport is everybody answer this question fast. No, we already discussed this one in previous question. The axonal transport can be either anterograde or retrograde. Coming to the next question, the neuron may get irreversibly damaged if exposed to significant hypoxia for 8 minutes, 2 minutes, 30 seconds, 15 seconds. The neuron may get irreversibly damaged if exposed to significant hypoxia. No, Dr. Santosh. हमारा जो axonal transport होता है, that is a bidirectional. From cell body to the axon and from axon to the cell body. How it occurs? See, a neurotransmitter is synthesized in Golgi apparatus. From there, it goes to the synapse. Okay, at synapse, they are released. A neurotransmitter will be released. And that empty vesicle again goes to the cell body for its refilling. Okay, so the axonal transport is a bidirectional. Is this clear, Dr. Sandesh? To reply fast, is this clear, Dr. Sandesh? Next is the neuron may get irreversibly damaged when it is exposed for hypoxia more than 4 minutes. So answer will be 8 minutes. Is this clear? Coming to the next one. Which is true regarding nerve conduction? All and none phenomena. Conduction is independent of amplitude. Propagated action potential is generated in our dendrite faster and undermyelinated fiber. Chalo, everybody answer this question. True about nerve conduction. Nerve conduction always and always follow all and none phenomenon. Conduction is dependent upon the amplitude. Propagated action potential is generated in initial segment, or you can say trigger zone. Faster and myelinated fiber. So answer is only and only A. Coming to the next one. A traveling nerve impulse doesn't depolarize the area immediately behind it. Why is it so? It is hyperpolarized, it is refractory, it is not self-propagating, the conduction is always orthodromic. This is a nice question, so everybody answer this question fast. A traveling nerve impulse doesn't depolarize the area that is immediately behind it. Why is it so? So these are two areas, suppose. So यहाँ पे अगर एक्शन पोटेंशियल स्टार्ट हो रहा होगा तो यहाँ पे रीपोलराइजेशन का सम फेज चल रहा होगा, and repolarization में we studied about the refractory period. ठीक है, हमने absolute refractory period पढ़ा था, relative refractory period पढ़ा था, and that what happened, the next action potential will not be generated. So immediately area behind हमारा किस period में होगा, refractory period में होगा. That's why the second action potential cannot be generated. So that's why nerve cannot go back. Okay, is this clear? Coming to the next one. Increase in threshold level on applying a sub-threshold level slowly rising stimulus is going to cause as accommodation, adaptation, refractoriness, or electrotonus. So everybody answer this question. Increase in threshold level on applying sub-threshold slowly rising stimulus is known as. We study about the two terminology. Sub-threshold level of a stimulus is also not going to cause generation of action potential because of accommodation, and supra-threshold stimulus is also not going to produce second action potential due to adaptation. So answer is accommodation. Is this clear? Coming to the next one. Increase in threshold level of a receptor when series of sub-threshold intensity is given. Same है, accommodation answer होगा. So skip करते हैं. Coming to the next question. This is also a nice question. Equal strength stimulus of which type is the least likely to produce a nerve stimulation? A, B, or C, or D? Everybody answer this question fast. Equal strength stimulation of which type is the least likely to produce a nerve stimulation? Everybody answer this question fast. We are seeing कि comparatively अगर B की बात करें तो in B we are seeing the rising of stimulus out here is slow. So slowly rising stimulus is having higher chance of accommodation and they are least likely to produce nerve stimulation. No, answer is the not C. यहाँ पे हम जो stimulus देख रहे हैं that is rising very fast. Okay. जबकि B की situation में stimulus की strength कैसे rise हो रही है? Slowly rising. And slowly rising stimulus has a higher chance of accommodation. Is this clear? Coming to the next one. Identify the ion based on conductance of neuron during action and potential generation. Sodium, potassium, potassium, sodium, sodium chloride, chloride, sodium. Everybody answer this question fast. Identify the ion based on the conductance of a neuron during action and potential generation. Everybody answer this question fast. Depolarization is due to sodium. Repolarization is due to potassium. So answer will be A, that is sodium and potassium respectively. Coming to the next one, absolute refractory period is due to opening of a calcium channel, closure of potassium channel, closure of active gate of sodium channel, closure of inactive gate of sodium channel. So this is also important question. So everybody answer this question fast. Absolute refractory period is due to. No, Dr. Sanju, answer is not B, it's A. Depolarization and repolarization. Depolarization is due to sodium influx and repolarization is due to potassium efflux. ठीक है, जबकि आंसर ऑप्शन बी is telling that is depolarization is due to potassium and repolarization is due to sodium. So answer is A, not B. Is this clear, Dr. Sanju? No. In absolute refractory period, any strength of a stimulus is not going to produce second action potential because in this, what happens? Inactive gate of sodium channel means our H gate is closed. Due to the closure of this inactivation gate, there is absolute refractory period. This point has to be remembered. Okay, is this clear? Okay, then, yeah, way to Dr. Apur. Coming to the next one. Initiation, excuse me. Initiation of impulse start in axon hillock plus initial segment, cell body or the dendritic tree. Initiation of impulse starts in. Yeah, the voltage gated sodium channel consists of a two gate, one at the outer side which is known as M gate, which is also known as activation gate. I guess you joined late, we discussed in the session only. And one gate is present at the inner side which is known as H gate or inactivation gate. Okay. On the basis of opening and closure of these two gates, there are three possible state of a voltage gated sodium channel is there. Resting state, open state, and the closed state. Is this clear, Dr. Divya? Is this clear, Dr. Apur? Is this clear? Initiation of impulse start at very true. Axon, hillock, and the initial segment, they are known as the trigger zone because concentration of voltage gated its channel out here is very high. Why? And that's why the threshold of these two segments are low. So they are the site of initiation of impulse. Coming to the next one. Myelination of peripheral nervous system is done by astrocyte, oligodendrocyte, ependymal cell, or the Schwann cell. Myelination in peripheral nervous system is done by myelination in peripheral nervous system is done by the Schwann cell. Oligodendrocyte is responsible for central nervous system myelination. Yeah, way true, Dr. Sandesh. Coming to the next one, true about the Schwann cell. Present in central nervous system, present only in a myelinated nerve fiber, form myelin sheath derived from surface ectoderm. True about the Schwann cell. They are not present in central nervous system. They are present in peripheral nervous system. They are present in both the myelinated as well as the non-myelinated nerve fibers. They form myelin sheath. This is true. They are not derived from ectoderm. So answer is only and only C. Is this clear? No, they are present in both the myelinated and non-myelinated fiber. This point is important. Do remember, everybody. Yeah, very true. Answer is C. Coming to the next one. Orthodromic conduction is an axon can conduct impulse in one direction. An axon can conduct impulse in both direction. Jumping of depolarization from node to node, print at which Ranvier spike potential. Orthodromic conduction is. Orthodromic conduction at the level of synapse is unidirectional. We studied this one. And excuse me. And at the level of axon, orthodromic conduction is bidirectional. Jumping of action potential occurs. Not only the depolarization occurs. Okay. So answer is only and only B. At the level of axon, the conduction is bidirectional, and at the level of synapse, conduction is unidirectional. Very true, Dr. Apur. Very true, Dr. Sandesh. Is this clear? Coming to the next one. Okay, that was the last question of today's session. आज के session में किसी को कोई doubt? If anybody has any doubt regarding today's session, you can ask. Hope you all like the session. If you like this session, do like and share. That is important. जितना ज़्यादा share करते हो, class share. And अगर अभी तक channel को subscribe नहीं किया है, तो subscribe कर लीजिए. Our channel is Unacademy Live in NEET PG. And do remember to press the bell icon so that you will get a notification of every upcoming classes on this page. Okay. Thank you. Regular मेहनत करते रहिए. You will definitely able to achieve your target. You will definitely crack your exam with good rank. Sorry. That's all. So let's crack it together, everyone. See you all in the next session. This is the code Dr. Nidhi. If anybody wants to join any paid course, you can use this to get additional 10% off. Okay. But do remember, we are going this unlock 20. When you use this code Dr. Nidhi, you will get additional 20% off. And this is valid till 30th. So if you're looking for the subscription, do enroll yourself right now so that you can save extra penny. Most welcome, Dr. Sanju. Most welcome, Dr. Sandesh. Okay, then see you all in the next session. Bye-bye, everyone.